if you, thank you. Okay, and I think Liz requested last week that I also include in here a little kind of diffusion MRI primer, and I think for the group here that makes sense. So what we'll do today, so the goal for the session today is that at the end you'll have some kind of fundamental understanding of diffusion MRI tractography. Not all diffusion analysis, but tractography in particular, because that's what we'll use to estimate the anatomical wiring of the brain. You'll be able, hopefully, to make some appropriate decisions about post-processing of those connectomes that you create using tractography, and that you'll understand kind of the formatting and basics of what needs to be input into TVB. So that we're ready to go for the next session for actually launching a TVB model. So the outline for today is we'll do a quick primer on both diffusion processing and tractography, talk a bit about the pitfalls of diffusion tractography and how that may play into your choices for post-processing of those structural connectivity matrices. I'll let Justin and Leanne take over to talk about what makes a good structural connectome. They'll also cover some of the functional features for optimization. So we'll talk a lot about the structural connectivity matrix inputs and things like that, because it constrains so much of the model, but we'll also touch on the functional aspects and functional features you can use for parameter optimization. And then at the end, we'll cover, well, not me, Justin and Leanne will cover the file naming and structure of TVB inputs. Okay. You may already be familiar to some degree with diffusion processing, diffusion analyses and whatnot. So I won't go into kind of like the nitty-gritty physics of it or anything like that, but I want to make sure that we all have kind of the fundamental understanding of how we arrive at these structural connectomes that we're using kind of as the input to the modeling software. So there are many possible workflows once you collect some kind of diffusion weighted imaging. On the right here are some kind of typical workflows or example workflows you could follow. You collect the data, just like functional MRI, there's some kind of artifact cleaning, there's some series of quality control and pre-processing that you can do. And then from there is where kind of the analysis can diverge, right? And in some case, you might know of some studies that looked at ROIs and did some RNI-based analysis. And you've also probably heard of things like fractional anisotropy and mean diffusivity, for example, so FA or MD, and these are measures you can use to kind of judge the integrity of white matter tracks in the brain. What we'll focus on today is tractography. And tractography is really the estimation of white matter tracks, in this case, in the human brain. Okay, so these are the target outputs for TVB. We're trying to create a structural connectivity matrix from using diffusion MRI tractography. And essentially what tractography is, if we boil it down to some very simple, I'll say 2 sentences about it, is one, we're modeling the directionality of fibers, and two, we're using those models to try to trace the fiber connections between regions. And so this I just grabbed from the website, the Human Connectome Project website. I believe this is a zoomed in picture of an axial slice of a diffusion image. And what you can see here, every one of these voxels along, this is probably a commiseral fiber of some sort, I'm just guessing. And then every single one of these voxels has a rendering of like probability distribution. This is the likely direction that the fibers are oriented in each of these voxels. And what tractography does, the two steps are you model the direction of those fibers per voxel, and then you send what are called seeds, and you step seed by seed, kind of like a path, and you trace where you are likely going to find these fibers. So this would be this white line. Oops, sorry. This white line is called a streamline. So you send like a streamline from point A, like our region of interest 1A, and you follow it along, and you see if it connects to region of interest B. And then you do that iteratively, thousands, tens of millions of times. You send lots and lots of seeds. You try to reconstruct lots and lots of streamlines. And what you end up with are very pretty images like this. Or you've probably seen them, very like detailed images of what streamlines may look like in the brain. Any questions there before I move on? This is bare bones basics. That's what I would like everyone to know about how we create a structural connectome. Okay. So, in terms of diffusion processing and tractography, there are some popular tools that you may already be familiar with. There's FSL. There's a really nice full package in FSL that handles both the cleaning of diffusion MRI and also the tractography steps. MR-Trix is a very popular tractography toolbox. There's also DSI Studio that I have not ever used, so I can't really comment on it, but I do believe that it's quite popular as well. What we have, so I would say supplied or offered with TVB is our own multimodal MRI pipeline for creating TVB inputs, I should say for creating TVB inputs that relies mostly on FSL routines. We forked this pipeline from the UK Biobank pipeline, so that's why it's heavily FSL based. It's the one thing I will say about this pipeline in terms of its diffusion tractography is that the steps have been validated using the CAC tracer data, and we'll go into this in a little bit more detail in a minute as to why we might want to do these kinds of validations in diffusion tractography. And what we've grabbed from here is just one figure from the pipeline paper. And this, I think this demonstrates a fairly generic set of steps you would do, whether you're using MR-Trix or you're using FSL for processing diffusion images. You do some kind of, this is kind of equivalent to motion correction, so eddy current correction and artifact correction. You might also do some image unwarping, distortion correction from the images because it can be highly distorted. You will do that model fitting for the fiber directionality. In this case, it's a multi-fiber model, so it's probabilistic model. And then you'll do the tracking itself, and that's the step of where you send seeds and you create these streamlines by stepping through many times to try to create a single streamline from one place to another, and then you do it over and over and over until you get many tens of thousands, millions of streamlines. And then you concatenate all that into a structural connectivity matrix. And in our case, we're after both the weights matrix, which is, we'll talk about this more too, is the probabilistic, is a normalized value of the number of streamlines running between any set of ROIs. We'll also extract from our pipeline distance matrix, as we talked also about how there's a space-time structure in TVB that we're after, and so the distances are important as TVB inputs. I'll leave here, and we've copied these, I think, into the Google Docs, some useful resources if you are looking to process your own diffusion MRI for TVB. there's a very simple primer on it on the Human Connect Home project's website that's really good. It's actually written in fairly lay language. There's FSL offers, as you may all know, really nice course and materials on essentially their whole package, and that includes the diffusion MRI processing. I would encourage you to go have a look at that. There's a lot of good background info there. MRTrix offers a fairly healthy set of documents for supporting you through if you choose to use MRTrix for tractography. And here I've dropped our own repository for our multimodal pipeline for TVB. It doesn't matter, I'll add here, it does not matter what you use to process your data for TVB. What does matter is that you have a good handle on what your processing does, and that you're confident that what you're getting out of your processing pipeline is nice, clean data. So the choices, often we leave the choices for cleaning data up to you. You know your data best. We have a few tools for you if you wanted to adopt them, but if not, keep your own tools and use your own tools because you know your data best. I'll comment a bit on kind of the pitfalls of diffusion MRI tractography. And this is because over the years, there's been a lot of discussion over how good is it for creating the structural connectome. Right? It can create all these really beautiful images, right? All these really detailed images of the, the anatomical wiring of the brain. And it has these really nice advantages where you get these weighted connectomes, you get kind of these capacity measures for how many fiber tracks might be running between any two regions of interest you might be interested in. You get an estimate of track lengths, which you don't get from the invasive histological approaches. And it's not invasive, right? So it's the only non-invasive way in vivo to collect this kind of information. But it has some disadvantages, and I think the main one is that it's highly inaccurate, and it's really susceptible to choices you make in tractography parameters. And this is really nicely demonstrated in this Meyer-Hein paper, where they challenged research groups to, using diffusion tractography, recreate a ground truth model, like a model they provide, and they say, here's an image, this is what the tracks should look like. Use your own routines and see what you can reconstruct. And when they I think they had something like 96 submissions, so like a lot of submissions across many different groups globally. And here is a very nice illustration of kind of some of the difficulties with tractography. So the first two columns here are deterministic tractography, and the third one is probabilistic. So in deterministic tractography, what happens is you end up underestimating or undersampling your tracks, right? You get highly specific answers, but not very sensitive ones. It's got high specificity, but low sensitivity. Whereas in probabilistic tractography, you have the opposite problem. You have a very sensitive method, but it's not very specific. So you end up with a lot of false positives. And you can see that it's very clear in these reconstructed streamlines that a lot of these are false positives. That's get this oversampling. And so, if a lot of us use probabilistic tractography, ourselves included, because it is so sensitive, but then there are some considerations as to what do we do about the output matrices if they are to some degree muddied by false positives. That's false positives. Yeah.
Talking about some of the post-processing choices we have to make for structural connectivity. So, straight out of the oven, I think SC isn't necessarily ready for TVB inputs. We have to do some kind of thresholding, some kind of data cleaning to make sure that it's all ready. So starting from the top left, I guess we'll talk about within subject normalization. And when we, at least for our workflow, when we perform, tractography, there's these two concepts of streamlines and probabilities. When you say, you seed region A with 1,000 streamlines, and you want to see how many of them end up in region B, streamlines is actually the count of the number of streamlines that make it to region B. So if you seed region A with 1,000 streamlines, 500 of them make it to region B, then your raw count for that connection between region A and region B, that would be 500. This idea of probability, on the other hand, is the percentage of streamlines that actually make it through. So again, if we seeded region A with 1,000 streamlines and 500 of them made it to region B, then our probability of connection for region A and B, that would be 0.5, 50%. With our approach, we go with probabilities, and that's more or less ready for input with TVB. But if you're working with raw number of streamlines, those tend to be really large numbers. And if you want to use structural connectivity matrices with those huge numbers, and you put them into TVB, you're going to have to adjust other variables like global coupling quite a lot to make up for that. So what you can do with streamline numbers is normalization across samples. So what you can do is you can look across the entire SC matrix of all your subjects, all your participants, and find the strongest connection, the highest, the streamline count, sorry, the connection with the highest streamline count. And what you'll do is you'll divide all your structural connectivity matrices by that highest count, and you're effectively just scaling down all your SC matrices to a more manageable number for TVB while keeping between subject differences. So that's the difference between probabilities versus raw number of streamlines. We'll talk about thresholding next, which Kelly mentioned is super important for managing these false positives, these potentially spurious connections. One of the approaches we can take is consistency thresholding. And this is kind of centered around the idea that connections that are very variable across different subjects are more likely to be false positives. So this approach basically ranks connections based off how variable they are across different subjects, and you can kind of choose a cutoff where if a connection is more variable than this value, maybe you'll toss it and you just set it to 0 for all subjects. So that's consistency thresholding. With consensus thresholding, you'll set a certain threshold, let's say 75%. And what you'll do is you'll zero out any connections that don't meet that threshold. So any connections where, let's say our threshold is 75%. If you have a connection where you don't have at least 75% of participants having that connection, you'll set that connection to 0 for all participants. So let's say this connection between region A and region B is only present in 50% of subjects. It doesn't meet our 75% threshold, so that connection is set to 0 for all subjects. Whereas if you have a connection from B to C and that connection is present in 90% of these matrices, that meets your threshold, and I'll just leave that connection alone. So you can see there's like two different approaches to thresholding and getting rid of these potentially false positives here. I'll move on to talk about symmetrization of the matrices. So in our approach, the DMRI doesn't let you really infer the direction of fibers. It just lets you know about the orientation. So we can't really build a directed matrix. We can't say, the connection from point A to point B is this value, and the direction of the connection from B to A is a different value. So what we do is we just average those two values and we create an undirected graph for input to TDP. And then, the next choice we have to make is, how do we handle NAND values? So the structural connectivity matrix that you input into TVB, it cannot have NAND values in there. So, classic data cleaning, you have to find a way to deal with that. And, depending on your question, depending on your project, depending on whose work you're trying to maybe compare your results with, you'll handle it differently. So one way is to, just remove the ROIs that tend to have more NAND values in them. If you think that the ROI isn't important for your analysis, You can remove subjects if it's only 1 or 2 subjects that are, tend to have a lot of NaN values in them. You can interpolate values. So say if you have one subject with a NaN value, but the rest of your subjects have perfectly fine values for that connection, you might consider interpolating that value from other subjects. And you can also, take inferences from existing literature. So if it's been established that a certain connection exists between two brain regions, and the connectivity strength is out there in literature, you might consider just manually inputting that into your matrix. So, these are all important choices that there's no one answer for, and it really depends on your research question and approach. And I think we're ready to move on to, looking at what, qualitatively, what does a good SC matrix look like, and what does a bad one look like?
We'll go to the next slide. Yeah, great. So these are just some examples of SC matrices and tract length matrices that we have from the TDB UK Biobank pipeline. So that pipeline has a quality control subpipe that we can use for the assessment of them. However, the judgments that we make may not necessarily hold for all the tools that we have. So take this with a grain of salt, that I'm just going to kind of walk through some of the things that we're looking for with these matrices. And another thing to consider is that they should often be examined in conjunction with various other processing steps along the way. So making sure that the T1 and DTI processing steps before making these look reasonable as well. So we have our good matrices on the left and a poor case on the right. So we see the matrix is the probability of connections between our regions of interest in the brain, and it's displayed in a logarithmic scale. And then below, for each respective matrix, we have a histogram of the connections. So we also have our tract length matrices, which shows the length of tracks that connects the various ROIs. And in our poor case, I guess, on the right, we can see that it's extremely sparse. If you're looking kind of at the scales, they're slightly different for the histogram, but the number of connections is very low in this poor case in comparison to the good case on the left, and especially within that interhemispheric quadrant. So the top right and the bottom left quadrants, we don't have the kind of horizontal line that you see on the good side within those quadrants of homotopy. So in general, they should be examined briefly, but in much greater detail if you do see that there are problems in the T1 or DTI processing that you may be able to address. Specifically for the poor case, just if you're interested, the problem with this was that the brain extraction was really poor, so we're not getting a good kind of capture of all the regions of the brain. So in addition to just visually expecting the matrices and making sure that they look reasonable, we can also use graph theory analyses to assess the quality and the network topology of the inputs into TDB. So with graph theory analyses, if you're not as familiar, we represent the brain regions as nodes, and then the connections between the brain regions as edges or links in the network. So in our example, for our connectivity matrices, the rows and columns are the nodes, and the matrix values in that matrix are the links that we're assessing. So we can compute a number of different metrics that characterize the graph. These are some examples of key ones that we usually look at to make sure that our matrices and our data look reasonable. So the first one is density, which is essentially the fraction of present connections to all possible connections. We can also look at degree, so it's just the number of links connected to the node. So in this example, for instance, the red node, the degree would be 4, as it has 4 connections. There's also various centrality measures that you can look into. For instance, betweenness centrality is the fraction of all shortest paths in the network that contain a given node. So nodes with high values of betweenness centrality participate in a large number of shortest paths. Just, I guess, to clarify what shortest paths are, it's not necessarily the tract lengths, but it's the number of steps between each of the nodes. So if you're going from red to yellow, the shortest path would be 1 to black and then 1 to yellow, so 2. Yeah, and there are a bunch of other metrics that you can also explore. But essentially we can use these metrics to identify either individual subjects or regions that are outliers. For example, if you're examining degree, you can identify nodes that have unusually high degree values. For instance, subcortical regions are particularly susceptible to false positives. So we can identify whether they're fully connected or overly connected to the rest of the network, and then decide whether we want to drop those regions from our analyses, or, as we kind of discussed, use thresholding approaches. So we can also use these to kind of assess the impact of the various processing steps that we use on the matrices. In addition to that, we can also kind of explore how the different network properties differ between groups if you have like a control population and a patient population. And you can see whether there's any underlying differences in the brain organization and connectivity patterns that can inform future modeling and kind of guide your research questions and approaches, or simply just to even assess the simulated data that comes out of TDP, which can be interesting as well to see if you can replicate certain features of the matrices that we're inputting. Okay, so that's graph theory. I think I'll go on now to some of the model fitting approaches. So we kind of discussed it last week, but I'll go over some of these metrics again. So a commonly used approach is maximizing the correlation between our empirical and simulated FC. So that's one approach, just taking that upper triangle and calculating the correlation. Previous studies that have used that approach, so fitting the static FC matrices, have been found to result in simulated network dynamics that are less realistic compared to when we use approaches that may maximize this idea of switching behavior or metastability in the simulated data. This switching behavior is essentially the max FCD variance value that we discussed. So I'll very quickly just kind of go over once more. I think we discussed it last week, how the FCD variance matrix is calculated, but essentially we take a sliding window approach where we get FC matrices for specific windows, and then we calculate the Pearson's correlation between the upper triangle of, for instance, time point 1 or window 1 and window 2. And that is essentially the first entry of T1, T2 for the FCD matrix. And then we take the variance of that upper triangle of the matrix. And in our parameter space exploration of all these different model parameter values, we want to pick the model parameters that give us that max value. Previously, this metric has been reported to be a proxy of brain fluidity and linked to cognitive flexibility. And thus, it can be a very interesting kind of potential target for model optimization. And then we also discussed previously, we could also minimize the KS distance between our simulated and empirical FCD matrices. So taking that upper triangle and then minimizing or making sure that they're as similar as possible. And then I also mentioned last week, we can explore how well multiple model fitting approaches kind of converge to identify your optimal model parameters, and we can kind of assess the quality of different approaches. And you can explore that depending on what you kind of want to replicate in your data. Yeah, I think that's all I have to say on that. So Justin, I think, can go over our last slide of kind of the file structures of the TBB inputs that we use.
Are there any questions about functional data? How we use it, how we might use it for parameter optimization? We have assumed here that because of the expertise of this group, everyone knows probably better than us how to get some nice clean fMRI data. This is really just what do you do with it after you've cleaned it to make it useful for TVB? And of course, the implicit part that we haven't said out loud yet is obviously the parcellation for these functional data matches the parcellation for the structural data, right? And that really is probably the only tricky part if you have two different pipelines for your fMRI and diffusion imaging. you need to try to get the data, the two data types parcellated in the same way.
Yeah. So TVB expects, certain inputs and structures for, the files that you input into it. In the black square you see at the bottom, that's an example of a directory for a certain subject and the, inputs that get it fed into TVB. So it really just takes these text files. And if you follow the link above the hyperlink above, it provides, TVB provides pretty good documentation about what it expects from its inputs. So there's certain keywords that you can file names. For example, at the very bottom, weights.txt, that is the file for your structural connectivity, and is probably the most important file. You can do without a lot of these other files, but weights.txt should be sufficient to start generating a model. The documentation also describes what the layout within each folder, within each file should look like. So for example, one ROI per row, what the column layout is like. And I definitely recommend you check that out. There's also on the GitHub a link to TVB data. So TVB provides a sample data set, with a bunch of different parcellations. And you can find these inputs in there as well. I recommend you take the time to open up these files, take a look at the structures, see what they look like. I guess I'll go briefly over the text files that are listed here. Centers.txt describes the locations of the regions of interest in coordinate space. Cortical lists out the ROIs and describes, is it cortical or is it a subcortical structure? Hemisphere, lets you describe, is it a left hemisphere or right hemisphere? Track lengths lets you describe, the track lengths. So it's, a lot of these are optional. Weights.txt is very much the most important one. And I will say that depending on your desired output modality, you may need more or fewer inputs than are listed here. So for example, if you want to output EEG data, you may need to provide information about your sensors, some region mapping, information about the surface. But if you're just going to go with our current approach, even just weights should be enough. And I think that's it.
That's it for our instructional part today. Any questions?